Good afternoon, everyone. My name is Jennifer Schultz. I'm the Senior Vice President of Marketing and Communications at OU Health, and welcome to our OU Health Research Roundtable. Throughout history, research has paved the way through some of the most medically challenging times. Thanks to the rapid deployment of research this past year, the world is finally seeing a light at the end of the tunnel with the December release of the first COVID-19 vaccines. Today, we have a panel of some of Oklahoma's most well-respected research experts to talk about the value of research, where it has brought us so far, and what is on the horizon in the fight against COVID-19. First, I'd like to introduce our panelists. Dr. Douglas Drevets is Chief of the Section of Infectious Diseases at the OU College of Medicine. He will talk to us today about the history of pandemic diseases, hi Dr. Drevets, and how research vaccine development has played an important part in getting past them. Welcome, Dr. Drevets, today. We also have Dr. Jennifer Holter with us today. Dr. Holter is a hematologist, oncologist in bone marrow transplant and cellular therapist with OU Health Stephenson Cancer Center and is very active in research on the OU Health Sciences Center campus. She will give us an overview of several of our current research projects.
It's a pleasure.
Thanks, Dr. Holter. Appreciate you being here as well. Thank you. Of
course.
And then we have researchers Dr. William Hildebrand and Dr. James Papin. They will be talking with us about their research projects. Dr. Hildebrand is with OU Health Sciences Center's Department of Microbiology and Immunology, and Dr. James Papin is a virologist in the Department of Pathology. Thank you for joining us.
Happy to be here. Thank you for having us.
Thank you very much for joining us today, and to our viewers. We are going to go ahead and get started. I know we have a lot of people watching and we have questions that are coming in as well, but we do have some questions we'd like to start with. Dr. Drevets, let's start with you. I'm wondering if you could give us a bit of an overview of what an infectious disease specialist does and what type of work that you do.
So an infectious diseases specialist is somewhat like a medical detective. We see patients, we deal with other problems, perhaps research questions, but in the patient arena, all we may know is that the patient has a fever, but many, many different kinds of infections can cause fevers. And so we spend a lot of time trying to figure out what the exact diagnosis is. And by diagnosis, I generally mean a microbiological diagnosis, meaning what is the infecting agent? Because they can all cause fever, weight loss, chills, and a whole variety of very similar symptoms and presentations. So it really takes a fair amount of intuition, training, and spending time to come up with the absolute right diagnosis.
So thinking about that, Dr. Holter, I know you are a hematologist, oncologist in bone marrow transplant. You also do research for OU Health Sciences Center and are involved in a broad range of COVID-19 research, from preclinical research through the Oklahoma Shared Clinical and Translational Resource Center to public health studies and more. Could you give us just a brief overview of the scope of research that's going on at the OU Health Sciences Center in COVID-19 today?
Sure, I'd love to. I think everyone needs to be very clear that none of the current vaccines or therapeutics that we have would ever have come to fruition had it not been the work of preclinical scientists that understood the mechanisms of the disease, or clinical trials that help us identify better ways to treat patients. So here at OSCTR, if everybody can see the slides, we have tried to put together all the COVID slides in one valuable resource where we'll be able to have all researchers see them, as well as all patients be able to interact with it. We have been looking at research with vaccines and vaccine adjuvants. We've been looking at trying to identify sugar molecules to suppress inflammation, and looking at encapsulated therapeutics so that we could try and deliver therapy to patients in different ways, including, like I said, liposomal encapsulation or different binding proteins, different antibodies to different portions of the virus. In addition, it's really important to have good public health policies. We all know the importance of wearing masks to decrease the transmission of the virus. But in addition, we've looked at the availability of trying to lengthen the time for some of the masks that we do use to try and lengthen the ability to do filtration in buildings, and trying to do AI and bioinformatics so that we can predict individuals that are going to be at higher risk because of ventilation in those buildings, and look at how we can utilize ventilators more effectively and make the number of ventilators we have not become such a resource that is constricted. We've also looked at the isolation impacts of children because of COVID-19 and the risk of maltreatment during the pandemic. And I think public health surveillance helps us identify hotspots, and helps us identify where we need to put our resources to try and prevent further spread. So we have studies with Dr. Raskob looking at those, as well as studies at looking at DVTs and venothromboembolus. Because this hit so quickly, multiple hospitals needed to collect information in a registry format. And in order to collect in a registry format, we had to get all the different hospitals together and have them put all their information into a large registry. We have participated in both the Discovery as well as the Stop COVID registries that will allow for that. And as a cancer specialist, we needed to know the impact when you're receiving cancer therapeutics of what COVID might have on your therapeutic process. And so we have the NCI COVID cancer trial that allows us to follow you through your cancer therapy for 2 years and look at that impact in multiple populations.
Now, Dr. Holter, I know you have some slides and we've had just a titch of a lag technically on the slides. So I just want to make sure that we're all looking at the same ones as you're kind of articulating these.
Should I start
if I'm
at the top?
Yeah, well, I'm thinking, I'm looking at this one here that's about the epidemiology studies for public health. And you were mentioning that just a bit earlier. Mm-hmm. And I'd love to hear a little bit more about, a lot of times when we think of research, we think of laboratory, but you're mentioning this public health research, and the slide that we have here, could you maybe explain a little bit about how that research is conducted? And you mentioned about people being in isolation and what have you, and I think that our viewers would be really interested in knowing a little bit more about that too.
Sure. So these studies look at, we've identified that there have been thrombotic or clotting complications associated with COVID. And so they're looking at incident rates of getting a clot in your arm or leg, getting a clot that moves to your lung called a pulmonary embolus, or having a stroke, and being able to look at surveillance data, meaning collecting data from different hospitals and groups so that we can, again, look at hot areas or hot populations that may have a higher incidence of these complications. We've also been able to look at the incidence of COVID-19 triggering some of these events by doing a longitudinal prospective study where patients have ultrasounds to see if these clots develop. Dr. Raskob has been instrumental in both of those studies.
Very nice. And then I know we have another slide here that kind of further articulates some of this research that you were describing. So if we can click to the next slide, please. This is the ICU National Registry that you were mentioning.
That's right. So this registry, called the Discovery Registry, along with the Stop COVID Registry, which I also think I have another slide, are both registry trials where when you are diagnosed with COVID and are put into the hospital and are treated on therapeutics that may be clinical trials or maybe standard of care, the data from your admission is then collected so that we can put all those into a registry and look for themes that we may be able to intervene on.
Dr. Holter, I think it's really interesting thinking about this idea of a registry, and I've heard you talk about this before. This is the first time we've all lived through a pandemic. Why is it so important for us to have a registry and to study this on an ongoing basis like this?
So because this is a new type of virus and we don't have as much of a knowledge base in how people are affected by it, we really have to pool all of our resources throughout the United States to collect the data and find themes. As a bone marrow transplant physician, we do this all the time. It allows you to identify small populations that may not be picked up at your institution, but when you put it across an entire United States registry, you can see them.
Very, very interesting to think about. Do we have another slide here that kind of further details this? Because this is really fascinating to think about. I know your body of work especially focuses on cancer, and this is really around cancer and COVID-19. Is that correct?
It certainly is. So as I was saying before, when you are diagnosed with cancer, that's scary enough. But when you get diagnosed with COVID in addition, we want to be able to help you still continue to get your cancer therapy, treat your COVID effectively, and look at the impact of both in populations. And so what this trial does is to evaluate that impact by just collecting regular data that you're going to be seeing with your own oncologist, no changes, just collecting it over 2 years to again see if we can see themes where we can do things better.
So I know that with your focus on oncology and that being your specialty, you're seeing a lot of cancer patients, but cancer patients with COVID. Do you have any kind of ray of hope or encouragement that you'd like to tell those patients or people that are maybe sitting at home that are worried about being immunocompromised?
So I think the ray of hope is that especially here at OU, we have a multidisciplinary team that gets together and tries to think about how there are differences and similarities in how we treat patients. Like if I have an autoimmune disease, it looks a little bit like what happens during COVID. Maybe we can find clinical trials to try and augment the therapies that we use in autoimmunity. Same thing with bone marrow transplant and cellular therapy. So I think that the good thing about OU Health is that we have all of those specialists, whether they be clot doctors, infectious disease doctors, cardiologists, transplanters, all working together to try and increase therapeutics. We also participate, if you go to the next slide, in multiple clinical trials that are available, including NCI COVID and the 2 active trials that allow us to get novel therapies that across the nation will be useful for patients.
So we are going to put this slide up at the end for people to really grab this contact information, but this is really a way for people to engage in COVID-19 clinical trials and that ongoing research that we know is so important. So thank you, Dr. Holter, for giving us that summary. I know, Dr. Hildebrand, that you are also involved in quite a bit of research. That's the theme of our roundtable today, and especially involving T cells. And I'm wondering if you could explain to us what exactly T cells are for the non-scientific viewer at home.
Well, in a non-scientific way, I suppose you could, the current vaccine is really activating B cells to make antibodies. And those antibodies are neutralizing the virus and working to keep the virus from infecting you. Maybe you could compare B cells to the army. You also have other components of your immune system, such as T cells. And T cells will help your B cells make antibodies, and T cells will kill the virus themselves. Maybe they're the Air Force. And so we didn't know when this pandemic started if we were going to need one arm of the immune system or a different arm of the immune system or both arms of the immune system. And my lab really specializes on studying the role of T cells in antiviral immunity, and that's what we've been characterizing.
So when you talk about antiviral immunity, is that essentially what a vaccine is trying to get you immunity to a virus? Explain a little bit how that works with the COVID-19 vaccines that are currently in use.
Well, what the current vaccine did was it took particular portions or segments of the virus, the nucleic acids that encode for the spike protein, for example, and they put it into a vaccine. And so they took a portion of COVID-19 and put it into a vaccine. They give you that vaccine, and then your immune system attacks the portion of the virus that they put into the vaccine. So one of the questions we were asking is, what portion of the virus should we put into the vaccine? And then which arm of the immune system should we try to mobilize or attack that portion of the virus? So yes, part of the virus is put into the vaccine, and you make a response to that portion of the virus.
So do we know how long something like T cell immunity will make you immune to COVID-19? Have we learned much about that yet?
Well, that's one of the things we've been tracking. It's quite amazing that we have this vaccine so quickly, because we're still learning how durable, and Dr. Drevets and Dr. Holter can certainly comment on this, but we've been trying to learn how durable is immunity to this virus? How long lasting is it? The common cold coronavirus, you have good immunity to it, but it's not really long lasting. This virus is more in the middle towards SARS and MERS. So we don't really know, but so far the T cell responses seem to be durable. And there seems to be some cross-reactivity with other coronaviruses. So it looks like we've got a fairly good T cell response that's helping your B cells make protective antibodies.
Now, one of the things that I've had some of my friends ask me about is if when they take the vaccine, when that moment comes, if they're basically getting a diluted virus, or if they're getting a different type of a protein to fight the virus. I don't know, Dr. Holter or Dr. Drevets, if you could maybe help us understand that a little bit better of what are we actually receiving if we're taking the Pfizer or the Moderna vaccine?
Thank you for that question. It's a really important concept to get a hold of. And as Dr. Hildebrand mentioned, there are more than likely, there are many different kinds of vaccines that are used to stimulate the immune system. Some are live, some are dead, and some are just parts of different infective agents. In the case of the Moderna and the Pfizer vaccine, they're what are called mRNA vaccines. So they are very, very simple. They take a small piece of the nucleic acid, or essentially the blueprints of the virus, and this is the part of the blueprint that tells the virus what kind of a protein to make, and they're called the spike protein. That's important because it's the spike protein that recognizes our body and that helps the virus invade our cells. So this vaccine, all it has is that little bit of the blueprint, and it's encapsulated in essentially a lipid balloon. Just think of it as a kind of like an ice cream balloon, so to speak. And that's all there is. There's nothing live in this vaccine. And it's a really a neat thing. There are other vaccines that are being worked on now. One is the AstraZeneca vaccine, which has been approved in the United Kingdom, which uses a live virus called an adenovirus to program our cells to produce the right kind of antibodies and T cells against the coronavirus. And then another company, Novavax, is studying a vaccine that uses a more of a traditional protein and then a chemical called an adjuvant, which stimulates the immune response. But the important thing to know is that in the vaccines that we have available now, and then also in the Nova vaccine, there is nothing alive in there, and you are not going to get the disease from the vaccine.
I think it's also important for people to know that when they hear that RNA, they think that it's the same as DNA, which is kind of the blueprint of how we make our bodies. And this process is not in any way going to change your DNA or put you at risk for changing your DNA, because it just can't get there. It doesn't have the building blocks at all. So it is a unique way that really had a lot of research at the NIH before we even got to the point of making this vaccine, for coming up with a way to kill and prevent coronavirus.
Dr. Hildebrand, you had another thought as well?
I just wanted to run something past the other panelists. When people have been asking me about this vaccine, the Moderna vaccine or the Pfizer vaccine, I've been telling people, and I just would be interested to see what your feedback would be from the other panelists, that conceptually, on paper, this vaccine is as clean and as straightforward and as simple a vaccine as maybe I've ever seen. It's very, very minimalistic. And that gives me some comfort as a person that's getting a vaccine that's been developed in Operation Warp Speed. I just wondered, do you feel the same way?
I do. I agree with you. There aren't extraneous proteins from cell culture. There's not a lot else in there. And so it does. It's, as you say, it's simple, it's stripped down, basically a couple of working parts, actually one working part and one inner part. And so it is a very neat concept.
I agree. And I think that the way I think about it is this vaccine is just helping your immunity to be extremely strong. It's just giving it a little bit of a push to identify what it needs to go after. It's like giving a little target and saying, hey, can you come and take a look at this? It's extraordinarily clean. It has it has the unique ability to just let your immune system do what it should be doing anyway.
Dr. Pappen, I know that you've been involved in a lot of vaccine research over the last year and in some projects that were a part of Operation Warp Speed. Tell us a little bit about what that's looked like this last year and some of the studies that you've been a part of.
Yeah, thank you. I think one thing to say is Dr. Holter made a point to say that a lot of this clinical research that happens due to the preclinical research that occurs beforehand. And Dr. Drevets touched a little bit on the Novavax vaccine. And here at the OU Health Sciences Center, we have had a long-standing relationship with Novavax and doing preclinical testing for them. And back in February, when this pandemic started, maybe not yet in the United States, but around the world, they were already looking at producing a vaccine. And they reached out, and we were able to establish some biomedical research laboratory models in order to look at the ability of their vaccine to produce an immune response. And that study is still ongoing, but what it did was contribute to their knowledge database that allowed them to move not only into phase 1 and phase 2 clinical trials, but now at the end of December, that vaccine has moved into phase 3 clinical trials. And hopefully in the coming months, once it's fully enrolled and we're able to gather data, that vaccine will also get emergency use authorization to help provide immunity to more individuals.
One of the things, oh, go ahead, Dr. Holter.
I'm sorry, I just wanted to point something out. I think one of the fears that many have about vaccines developed through warp speed is that they're new and they haven't been studied well and there isn't good data for them. And I think that what Dr. Pappen just said is very integral for everybody to understand. None of the vaccines that are developed were pulled out of the blue. There was significant preclinical work that was already ongoing in preparation for something like this to occur. And we just were able to tap into it and then develop lots of vaccines at a faster rate, whether they were to work or not. But that population of data and work has been done by investigators throughout the United States and world and was already there. So there were years of data that were behind it.
Yeah, that is very well stated, because a lot of people are concerned about the fast track of vaccines, but many of these vaccines are built on platforms that have been studied for a very long time. In fact, you look at even the new platform that we have emergency use authorization for right now in the Pfizer and the Moderna vaccines with the messenger RNA, they've been looking at that for over a decade. That's been studied for a long time, and we've known about messenger RNA and how it works in the cell for what even much longer than that. It was shortly after DNA was discovered that messenger RNA was thought of as a concept and then subsequently demonstrated that it existed. And so our understanding of how these things work and how they're safe for the body, that has happened for a very long time, and it not just something that came out of the blue over the last year.
And we have therapeutics that take advantage of that in cancer right now. So it's not like this mechanism was new. It just was new for something that came out of the blue.
Basically taking a very large body of scientific research and applying it to COVID-19. Is that essentially how maybe I should think about it and my friends should think about it?
I think that's exactly what it is. And it brings to the forefront how important multi-subspecialty and linking preclinical and clinical scientists and research together, and how quickly you can move when you have that base already there.
So the term emergency use authorization has come up quite a bit. And I know that's a question that a lot of people in my life that, aren't involved in healthcare want to know, what does that mean really? And how is that different than if it was authorized without an emergency use? And can I have confidence in the vaccine when it's emergency use authorized? I'm wondering if one of you might be able to help us understand that a little better.
So my understanding of the process is that the emergency use, it still went through the vaccines, the Moderna and the Pfizer vaccines. They still went through the usual phase, what are called the phase 1 or the preclinical, then phase 1, phase 2, and phase 3 trials. The phase 3 trials are the ones that involve tens of thousands of people. The Moderna trial involved around 40,000 individuals, as did the Pfizer trial. The one that's going on by Johnson & Johnson right now involves somewhere around 40 to 50,000 individuals. So these are large numbers of people. And these are the trials where the safety and the efficacy of the vaccine are looked at, in a sense, thousands and thousands of people, compared to a placebo, which is basically a fake. In this case, in some cases, it's a shot of saline. In some of the trials, it's a completely different vaccine to a different disease. So the safety evaluation was essentially the same going through this. I think the only difference is that the manufacturers did not have a year's worth of data or this long-term data. And of course, that is not practical to obtain during a pandemic. If we stuck to that before you gave full approval, in a sense, and you thought that was important, you know, just in the United States, several 100,000 individuals will die in the next year if you didn't start vaccinating now. And so it makes sense to use this emergency use authorization, but I think we can have confidence that the vaccines are indeed safe and went through the same safety protocols and the same vetting protocols, the only difference being length of time afterwards.
And I think the other point is you have millions of healthcare workers that have been vaccinated and that are adding to that data so that you know that this is a safe alternative for you. As a practitioner that has seen some of my patients get COVID and not make it, there is no question in my mind that I wanted to participate in supporting my fellow Oklahomans and my fellow Americans in trying to prevent this from going to individuals at greatest risk. And I've had, in fact, I had my second vaccine today with no symptoms whatsoever and feeling fine. So I would tell people, you must understand that your risk associated with COVID could be great, and the vaccines have been very safe and vetted through the FDA in a very good fashion.
Yes, I think that's a great point, Dr. Holter. And I also had my vaccine just about an hour before this meeting. And so everybody is welcome to watch me and see if I either pass out or grow a second head, but more than likely I won't, because this was my second dose and it didn't happen with the first dose. So.
Excellent. Dr. Hildebrand, I know that this past year you launched a research collaboration with Pure MHC, which is an innovative biotechnology company to work towards development of a vaccine. Can you talk a little bit about that project and sort of where that research study is at?
We got into the game early in February or March of last year, working with Dr. Papin and talking to Dr. Holter and Dr. Drevets, but you could see with the COVID-19 vaccine and Operation Warp Speed how public-private partnerships came together. It was an amazing coalition of people that came together to make this vaccine possible. Nothing like this has ever been seen where a vaccine came out this quickly. No one predicted really, except for the companies that were doing it, that it would happen this quickly. And so we saw this happening. We actually did it as fast as anybody. We said, what's the best way to begin to investigate this? And it wasn't to wait. It was to gather your resources, find people that were interested and wanted to approach this, and that's what we did. I would say that Moderna and Pfizer did an outstanding job, and they have the right solution. And we didn't have necessarily the right solution in this instance, or a solution that was better than theirs. But I think that The success here is that everybody attacked it. Nobody stood on the sidelines. Everybody got into the game, and somebody came out on top with a vaccine. And I think that's what we were all after.
And it sounds to me like when you're trying to move towards a vaccine that quickly, all of those different ways in which you're researching, finding it, is collectively important to get to efficacy of one or more vaccines. Is that correct in my understanding of that?
I think I see everybody's head shaking. So I think that, you know, I mean, if you were John Grisham or someone who wrote novels, you could write such a story on this. There's Drew Weissman and Dr. Carrico at Penn. They almost lost their jobs because they worked on RNA as a vector for moving things for gene therapy. They're both going to win probably a Nobel Prize now for this vaccine. So you're going to see Dr. Drew Weissman and Dr. Caitlin Carrico probably win a Nobel Prize for this. Yet it almost ruined their careers to work on RNA, but they persevered. You saw a company, Moderna, take RNA and we'll learn how to package it, manufacture it, code it, and deliver it, but it never worked for its intended purpose. Then you saw Dr. Mike Diamond, a physician scientist at Washington University, use it for Zika, the first time for infectious disease, and develop a successful Zika virus vaccine. That all laid the groundwork for when the coronavirus came out, that they plugged a piece of the coronavirus into RNA, put it into the Moderna Pfizer packaging system, and delivered it quickly. It's a novel. I mean, you in the government, in the private industry and government and researchers all got together and combined the parts. So that's what we tried to do here. We tried to contribute however we could, working with the university, the company, and I think that's why you have a successful vaccine, is because people teamed up.
Well, and Dr. Hildebrand, that just really is inspiring to think about. It gets me really excited. I think back to Dr. Holter's comment around how mRNA had been studied in a lot of other ways for years. So that the type of study and data that's occurred, we don't know what that might lead to for future breakthroughs in other areas of healthcare, it sounds like. How does that work within the research community, that data set and collaboration, and maybe a vaccine study that was today that could maybe be utilized and break through something new 10 years from now? What does that look like?
I'll say real quickly, and the others can jump in, you couldn't have scripted this. The reason we have this vaccine, in my opinion, and the others can contradict me if need be, is that the American taxpayer has gotten behind science so strongly and so well and funded so many things that you had Zika virus investigators, people with gene therapies, companies making solutions to gene therapies, nanoparticle people making lipids. You had such a diverse group of people, all funded by, much of it funded by NIH, some of it by industry, come together to do this. You couldn't have scripted this, I don't think. I think that the reason we've been successful is because so many people were working so hard on so many different fronts.
I agree with that. And one thing that people have to know about research is the vast majority of it, you think you're going in one direction, but all of a sudden other avenues open up, and this may be years after you've done your fundamental work. For example, I've been involved in infectious diseases research for almost 30 years now, and I've been studying how bacteria invade the brain and the immune response in the brain. I didn't know even a few years ago that was going to take me into a specific T cell population. And then this last fall, Dr. Hildebrand and I wrote a grant together about a T cell population that was just discovered by other people a few years ago. And I happened to find it in the model that I'm using in the brain. And Dr. Hildebrand knows how to look at parts of it. I know how to get it there. And so it was a great team up. But you could not have made this up even a few years ago. So research, much of it builds on itself, but in a very unscripted way.
Mm-hmm. And I know research and the, looking at research funding and grants and all of those different pieces, that helps to accelerate sometimes the speed of reaching a breakthrough. And that's, I think, what we saw with Operation Warp Speed. I know there's a lot of collaboration across Health Sciences Center around this. And Dr. Pappen, you were doing a research study, and I think it's still ongoing, around antibodies of previously infected COVID patients, and that you're working with OSCTR on neutralizing the antibody study. Tell us a little bit more about what that is. I think antibodies and understanding if you're immune or not is something a lot of us are curious about, but maybe don't understand fully.
Yeah, so antibodies are special proteins that Dr. Hildebrand talked earlier about, B cells, produced by B cells, part of your immune system. And these special proteins are designed to bind foreign entities that enter your body. We call them antigens a lot of time, and antigens are often pieces of infectious disease. And the role that antibodies usually play in viral infection is to help block the infection before it ever occurs. And a term a lot of people might have heard recently, or at least over this pandemic when it comes to COVID, is the idea of neutralizing antibodies. And neutralizing antibodies are those antibodies that can bind the infecting agent, such as the virus that causes COVID, and keep it from infecting cells. So we've heard a lot of talk about the spike protein on the SARS-CoV-2 virus, which causes COVID. And that's the protein that's responsible for binding to the cell and allowing entrance of the virus into the cell. There's a specific part of that protein called the receptor binding domain, and that little portion of that protein is what attaches to the cell and allows entrance. Well, neutralizing antibodies bind at or around that binding domain, and those are very important for keeping infection from occurring. So one of the things that we're looking at is we actually take authentic SARS-CoV-2 and we take the portion of the blood from patients that have recovered from COVID and then study the existence of those antibodies and their ability to block the virus from entering the cell. So we do that in collaboration. This is really an OSCTR project. We just have the specific expertise to perform that assay in order to look at that and provide them with those data.
And I was going to add that the OSCTR is doing a biorepository, so we'll have samples for investigators like Dr. Pappen and Dr. Hildebrand so that we can further study this. In fact, we're changing or augmenting the biorepository to collect information after people get vaccines so we can look at the neutralizing antibody differences between those that natively got COVID and those that got the vaccine and followed over time, which bespeaks the length of immunity that one might have with each of those events, whether it be primary COVID versus vaccination. So we'll be collecting data in the biorepository and samples in the biorepository that will allow those investigations to continue.
Dr. Holter, that makes me think a little bit too about some of the modern technology that is being utilized around collecting data and for the purposes of research. And I know that there's studies that have involved the use of a smartphone, and we have a slide that sort of illustrates that I think we should pull up for everyone. And what I'm wondering is what are other significant COVID-19 kind of research pieces are you hearing on the horizon, and how does this data collection kind of work as far as discoveries go? I think I've got you on mute right now.
I think I'll try it now. Let me start with the COVID app. Dr. Moxley, who is one of the gynecology oncologists over at the Cancer Center, came up with this innovative trial that's funded through the NIH that allows for patients that are undergoing cancer therapy to receive a mobile app that they can either place on their phone or if they don't have a smartphone, they will receive one. And it checks symptomology throughout the week. And if after questioning about certain symptoms, it triggers that maybe you should be tested for COVID, you will be contacted and a test will be set up. If you do become positive for COVID, subsequently, you'll receive an oxygen monitor and a pulse monitor, and we'll be receiving phone calls so that we can determine, and questionnaires, so that we can determine if you need to be treated at home or whether you need to be treated in the hospital. I think this is one of those neat opportunities that we were able to identify, and has changed with COVID. We would never have been able to institute this kind of trial so quickly without NIH funding. And it allows us to think of novel ways of treating our patients, which I think is great. The next slide I think you have is a couple of trials that are on the horizon that we think will be available, that include using, as I said earlier, there are active trials, active 1 and 2, as well as active 4, that utilize what knowledge from other autoimmune and cellular therapeutic bases in order to treat COVID. And they're putting these drugs into clinical trials so that we can try and treat patients much, much better.
Dr. Holter, we have contacts up right now for inclusion in clinical trials for COVID on the Health Sciences Center campus. So just so I'm kind of understanding this correctly, is this for people that currently have an active COVID diagnosis, people that have recovered from COVID, or what type of individuals are we engaging right now in these clinical trials?
So for the Stephenson Cancer Center, they're primarily patients that have a diagnosis of cancer and then have been diagnosed with COVID or have cancer and would qualify for that mobile app that we were speaking of. For OSCTR, there are multitudes of studies that we're using the biorepository for. So if you have COVID and you do not have cancer, you do have cancer, anyone who has a diagnosis of COVID within a week can contact this number and be involved in that biorepository. If you are looking for therapeutics, I would say the Stephenson Cancer Center number would be useful to try and get you to the right person for any of the novel therapeutics that are coming down the pipe, because active 1, 2, and 4 are for patients that have cancer or don't have cancer. So they would be for all comers.
Excellent, thank you. We have just a couple of questions left and then we're going to wrap. One of these questions, I think maybe Dr. Drevets, you could help me out with, and it's really around the concept of herd immunity in the vaccine. And the question from one of our viewers is, if you're someone who is in a vulnerable population and you get vaccinated, can you start moving around normally again, or should you wait longer until there's some sort of herd immunity?
Thanks for that question. That's an important thing to think about. A couple of comments about herd immunity. That's the idea that when enough people in a particular population become immune to an infectious disease, that the germ no longer can find new individuals to infect. That's the concept of herd immunity. And before vaccinations, all that we had was natural immunity to infectious diseases as to how we overcame them. That said, herd immunity is a good thing, but it's not a perfect thing. Think about many of the infectious diseases that were around, certainly earlier in my lifetime, such as polio. There were still ongoing polio epidemics in the 50s and the 60s until vaccination started. Things like smallpox, which killed hundreds of millions of individuals over several centuries, and it probably wiped out about 90% of indigenous Americans in North and South America in the 15th century. That didn't end until the 1970s when it was eradicated by vaccination. So I just don't want people to think that herd immunity is perfection. It's a good thing, but it's not a perfect thing. In terms of the question that you asked, if somebody is in a vulnerable population, they absolutely need to get vaccinated. This is the best way to protect themselves. The problem is that when you're in some of these vulnerable populations, we don't exactly know how well your immune system will respond to the vaccine, and will you develop really strong immunity, or will it be somewhat weak, so to speak? So at least for now, we're advising people to continue to wear masks after receiving both doses of the vaccine. And we should do this either until we have really good data from the manufacturers that the vaccine will work in people just like the one who asked the question, or until we see a major drop and decline in the level of community transmission so that we can safely avoid the mask. Remember, the mask is not perfect either, but it's the best tool we have now to prevent the infection from one person to another.
So thank you. That's a very, very good clarification for all of us. I think in closing, we'll just go through each member of our roundtable. I can't thank you enough for taking the time. This has been hugely informative. Perhaps closing remarks from each of you on COVID-19 and research and what we see on the horizon. And so maybe starting with Dr. Hildebrand, any closing remarks from you?
I would probably just reiterate how exciting it has been to watch this vaccine emerge in the last less than a year, and to see how people have come together to weather the storm and now implement a way to defend against this virus. And I would encourage people to learn the lessons. We pick up the lessons we can learn from this this struggle we've had, and to apply them in the future.
Excellent, thank you. Dr. Holter, any closing remarks from you?
I just want to echo what Dr. Hildebrand just said. The amount of interaction between both preclinical and clinical, as well as multi-subspecialty care, has been profound, and I hope is a model for the future. My dad is an Army nurse, and he was itching to be a part of the vaccination so that he could help his fellow Americans. I would like for everyone to see this to be just one more avenue for you to help your fellow American, help and get involved in clinical trials so you can help yourself and help the person sitting next to you. And please mask.
Thank you, Dr. Holter. Dr. Pappen, any closing remarks?
I'll just further echo those sentiments and say that, both scientifically and socially, let's take what we've learned over this past year and keep it close, because really infectious disease is one of the greatest threats to mankind, and we need to make sure that we stay diligent in our scientific research and our social responsibility so that we can, one, overcome this current pandemic, which we are still in the middle of. And while we have vaccines and the light at the end of the tunnel, there's still some ways to go. And second, if there is a circumstance where this happens again in our lifetime, or in our children's lifetime, or our grandchildren's lifetime, let's have that data and that understanding be there, just like it was in the past, so that therapeutics and vaccines can move forward even faster than what they have now.
Thank you. Dr. Drevets, any closing thoughts?
Yes, a little bit of a history lesson. Many of the things that we are living through now were, in a sense, invented for the very first time in history because of a pandemic. For example, quarantines. We've all talked about quarantine, and people have heard about this. That was developed in the 15th century because of a bubonic plague epidemic in Venice. The idea of vaccination, that came about by Dr. Edward Jenner in 1792 against smallpox, another pandemic organism. And also the whole science of epidemiology. This is how we track people. And it doesn't just apply to infectious diseases, it applies to cancer, high blood pressure, diabetes. The whole science of epidemiology was founded by a gentleman, Sir John Snow, in 1852 in London, when he was investigating a cholera outbreak. So it's been hundreds of years of work of pandemics that go up to leading to what we have now. And so let's take what we're learning now also and build for the next generation.
Thank you so much. I can't thank all of you enough for taking the time on a Friday afternoon to educate us and help us understand how research is really our hope for the future with COVID-19 and for many other things. You know, when I think about healthcare and advances in healthcare, research is the key to all of it. And we know all of you are very engaged. It's the work of your life, and we can't thank you enough because we know ultimately it takes care of us. So thank you so much today. And we just hope that all of our viewers had some great things to take away. If there's any other questions or things that you have, feel free to reach out to us here at OU Health, either through our direct message on social media or to our media relations team. And have a great afternoon, everyone. Thank you.